This tutorial will walk you through the online learning space of Cell Academy. You can log in through our website, cell.academy/login. You can also register for demo access by filling out the form on that website. If you log in through our website, you are redirected to a login page. Another way to access is directly through the software. When you open the software Steve, there's a little cloud icon on the bottom left. By clicking on the cloud, a login box pops up, where you can then enter the credentials that we send you when you sign up. The learning space then opens up. This is where you can access the different lesson plans. If you're a user of the 3D Cell Explorer microscope, you will also have access to the protocols. Let's click on the cheek cells, for example. When you open the lesson plan, you have different files. There's always a worksheet, which you can then open, and it gives a short summary of what you are to expect from this lesson plan. It gives the clear learning goals. Some background information is provided to teachers. There are then some questions to ponder, which you can go through with your students for an interactive class discussion. This is where you find the worksheets to either print out or send to your students. It explains step by step what the students will have to do with the learning space material. For example, in this one, the first step is to open the vol file 1. Vol files are the 3D cell data. Here you can see that back in the software you can open the vol file. It opens in Steve and you automatically have a 3D render that is generated by the software. This is just a rough estimate of what you can expect. You can then add your own colors, which are defined by the refractive index of the different organelles in the cell. For example, the nucleus here, when you focus on it, you can choose a color like pink and just make a rough selection of pixels. You will then see any other pixels which have the same refractive index will then light up. You can make your staining more opaque and get a beautiful 3D render of your nucleus. You can then travel through the slices with the left mouse button or using the slices slider beneath. If we return to the cloud, we also provide stain files in case you do not want to do the staining on your own. You just have to click on it and it applies a pre-saved digital staining that we have done, which then helps you see a 3D render of your cell. You can see an overlay of the staining on the refractive index map. You can also remove that if you just want to focus on the raw data. Another useful tool for some of the exercises is the ruler tool, which you can see up here. By clicking on the ruler icon, you just click on one edge of the cell and the other, and it gives you the measurement in micrometers. You may have also noticed these numbers in the staining panel, which represent the volume of the staining. So this color in blue represents the volume of the nucleus here in blue. With these tools, the students can then answer the different quantitative questions that we provide in the worksheet. At the end of each worksheet, you also have the alignment with education standards, NGSS, IB, and the national curriculum. So as you can see, it's very simple, intuitive, and fun for the students to use.